Welcome, everyone. This is Bio460, Experimental Methods in Advanced Genetics. I'm very happy to see you all here in this class. Although we are going to perform the class online asynchronously, I will teach you the gene cloning procedure, and also I will deliver the concept and the fundamental knowledge in gene cloning through the class. And first of all, let's go through the syllabus, because this is very important. That would give you the information how to go through this class. Okay, so let me share the screen. So this is the syllabus, and I post this syllabus in the Blackboard already. So you should be able to find the document in the syllabus. And this is my name. This is Dr. Shen, and I'm the class instructor for Bio 460. And for this class, and this is my email, and as I mentioned, this is online synchronously. We will perform the class online synchronously. And this is my email address. At any time during the class period, you will be able to reach out to me through the email anytime. Okay, if you have any question, and you can reach out to me through the email. And the course objective, basically, this class, we will go through the book Gene Cloning and DNA Analysis, right? So basically, I would give you all the important concept and knowledge for the gene cloning. In the meantime, I also will teach you how to design the gene cloning experiment. And so that comes down to this assessment for this class. As you can see, for the evaluation for this class, we will have the final exam. That's 40% of your final grade. And also we have the lobster 30%. As you know, we are not going to perform the laboratory, and so that's why we adopt this lobster, and you will need to perform those lobster exercise. And later on in the syllabus, I will tell you the progress of the exercise, and this will be 30% of your final grade. And then on top of this, we have this 30% here, 20% that will be the lab report, because you will be required to submit a gene cloning project, and that's the reason why you take this class. And also that will be the knowledge I'm going to teach you how to design a gene cloning project. And also you will also need to submit a library research project report. Is a 10% of your final grade. So 20% will be the experimental design of a gene cloning project, and 10% will be like a library research, the literature research report. Okay, and 40% final exam, 30% will be lobster. So this is very important, lobster. And another thing, the next thing is that if you have any disabilities, you will need to submit a document so that I can accommodate. So for example, to accommodate you for a longer period of time for the exam, all right? So that will be very important if you can submit a document. And also the academic honesty statement. So basically, everyone is responsible for being aware of what constitutes cheating and the plagiarism, and for avoiding both. And cheating of any kind will result in immediate failure of this course. Okay, so it is very important, all right, although it's online, but still it's very important that you will follow the procedure, and also to avoid the cheating and the plagiarism. Okay, and then as I said, this is online asynchronous course. However, I will provide the several office hour, and then if you have any time, if you have a question, you are welcome to come to the office hour or email me. All right. So if you're going to email me, make sure in your subject line you have to put a bio 460 and then your last name, so that I will be able to know this email is from the class. And then here is a list of the date and the time that will give the office hour. So for example, January the 3rd, between 10:30 and 11:30, I will give the in-person office hour at the room 6S143, which is the biology office. And then January 5th and 7th, between 10:30 and 11:30, then I will have the Blackboard office hour, which will be on the Blackboard at the Collaborate Ultra. So you have to come to the Collaborate Ultra, then you will see me on the Blackboard, the class Blackboard. And then January 10th, 12th, and 19th and 21st. So these are the dates and time that will give the office hour. And on top of this, you are always welcome to email me at any time. And then I will respond to your email, or we can set up, or we can arrange the appointment and to discuss the class or class materials. Okay, so that would be the office hour. And then for the syllabus here, so as you can see, and there are 4 different columns. The first column will be the date, the number of the class, and the date. And the number of class here, that will refer to those video that I post on Blackboard. All right, so you will refer to the class 1, then you go find the video, class 1 video, and then to study the class 1 video, and then class 2, class 3. So all the video has been labeled according to the class number. And then the second column will be the topics of that class. And then the column 3, that will be the pages of the topic in the book. Remember that we're going to use the book, our class basically based on this book, Gene Cloning and the DNA Analysis, an introduction by the T.A. Brown. Okay, although I put the 7th edition, you can use 6th edition, 8th edition, right? So it doesn't matter, because the material, the content will be almost the same. And then this is the third column. And then the last column, which is very important. So you have to find this column here. Basically, that will be the topics for the lobster exercise. You will go to the lobster and then to perform the exercise based on these progress. So for example, class 1, then you will need to complete this 4 lobster. And class 2, you are required to perform these 5 different lobster exercise. And then class 3, that'll be these 2 lobster exercise. So all these, the column 4 here, basically just to give you the guideline where to what topic or what exercise you need to perform for the lobster. And remember, the lobster exercise will be 30% of your final grade here, 30% of your final grade. So it's very important that you will need to perform those lobster exercise. And you do not need to follow this schedule to perform your exercise. If you have time, you can even perform all the exercise in 1 week, because I have already opened the exercise since Monday, January the 3rd, and then all the way until January 22nd. Okay, January 22nd will be the last day, the deadline for you to complete all the lobster exercise. January 22nd, Saturday, midnight. You will need to complete all the lobster exercise by January 22nd, Saturday, midnight. Very important. And then for the whole class, as you can see, we will have no quizzes. We don't have quiz. We don't have midterm exam. However, we will have the final exam. That will be on January 24th at 9:30. Okay, so the exam will be open at 9:30. You'll be able to enter the Blackboard and perform the exam. Okay, so 9:30, that will be January 24th, and the final exam, that's 40% of your final grade. All right, so 9:30, very important. And then the final exam will be everything in the video in that I post on the Blackboard, and also the books. And also on January 24th, you are also required to submit your laboratory report, all right, laboratory report, and also the library research report, okay, by 10 a.m. And then, of course, every 1 hour delay of the report will cause a 5 final grade point subtraction. That's you'll be penalized, okay? So make sure you have to submit your reports, both the laboratory reports and the library research report by 10 a.m. And each of you will be assigned one specific topic for the laboratory report, and also one library report. So that will be done by January 7th. I will give you the assignment. Each of you will be assigned one project and also one library research topic, and so you will be able to perform the laboratory experimental design and the library research for the next 2 weeks, because I will announce that by January 7th, and the reports are due January 24th. And remember that January 21st, okay, I will give you a final review during the office hour, all right, during the office hour, January 21st, as I show you here, January 21st, that will be between 10:30 and 11:30 on the Blackboard. So if you are interested, you are welcome to come to the Blackboard during that time for the final review. Okay, so that's basically the information for this class. If you have any question, you are welcome to come to visit me during the office hour, or you can also email me about the class. Okay. And now I'm going to give you some introduction about the class Blackboard. So when you log in, you probably will be directed to this page that you will see all this Lobster simulation. And all this simulation have been opened, okay, from the January the 3rd until January 22nd. As I mentioned, the deadline to complete all the Lobster will be January 22nd, Saturday, midnight. You need to complete all the Lobster, okay? All this Lobster. So when you look into the class Blackboard, then you should be able to you'll be directed to this page, and then you will see all the Lobster simulation. And then also this, on the left panel here, on the panel on the left side here, you will see the Lobster simulation. If you don't see this page, you can always find the Lobster simulation and click this, that will direct you to this page. And all the class material will be posted on content. So if you want to find all the class material, you need to click this content, and then go to the content page, and then you will see all the class materials, including the syllabus and the class video. And to come to the office hour, you have to click the collaborate, all right? You click the collaborate, then that will lead you to the course room, then you'll be able to join the course room. So basically, this is the introduction of the class Blackboard. We just went through the syllabus. So for this class, then first of all, gene cloning project, we have to know what is the gene, all right? So gene, each heritable property of an organism is controlled by a factor called a gene, and that is a physical particle present somewhere in the cell. And the gene basically has a structure that we have the promoter, open reading frame, all right? And that's the open reading frame. That's basically is the gene sequence for a gene. And the promoter, that's the where the ribosome will bind to translate the messenger RNA into the protein. And also in the end, we have this stop codon to to uh, um, to conclude the gene. Okay, so that's the size of the gene that you need to know. Later on, we will go through that in detail as well. And the biotechnology, basically gene cloning technology, and it's a recombinant DNA technology, and also called genetic engineering. The recombinant DNA technology is because we have to we have to take a piece of the DNA sequence, and this DNA sequence contains the gene, and then we put this piece of DNA sequence into another piece of DNA sequence. Then we can introduce this whole recombinant DNA molecule into the cell and to either reproduce or for the production of protein. And so this whole process, it's the biotechnology, and it's called gene cloning, and it's a recombinant DNA technology, right? So this picture just explain what I just said about the recombinant DNA technology, all right? So basically, you have this you collect the DNA from any organism, and then the genomic DNA that you collect from an organism. Then from the genomic DNA, then you have a piece of the DNA of interest, and that will be the gene of interest. Then you can try to either use the restriction enzyme to cut that piece out and reproduce it, or you can reproduce by using the PCR to reproduce that piece of the gene of interest, and then give you a piece of DNA sequence. In the meantime, the scientists that they already isolate a plasmid, which is a circular DNA that can be reproduced in the cell. All right, so they isolate the plasmid DNA, and then that can be a vector. Vector basically is a vehicle that can carry the for a piece of a foreign DNA. All right, so we use this plasmid as the vector, and then we introduce this piece of sequence into the vector, and this procedure is called cloning. All right, this procedure is called cloning, and then you produce this recombinant DNA plasmid, and then you can introduce this plasmid DNA, the recombinant DNA, into the cell. All right, into the bacteria cell, and then after that you can either reproduce this DNA sequence, the vector, the plasmid, all right? And then you can study the function of the specific gene. Or you can use the bacteria cell, all right, to produce a protein. And then the protein can be used by the human. So here is the example of some of the pharmaceutical product of genetic engineering. So it can be the alpha interferon, right? So it's a therapy for the leukemia, all right? They produced by the E. coli and the Saccharomyces cerevisiae, all right? So basically, you through the gene cloning, then you introduce this recombinant DNA into the E. coli. Then the E. coli bacteria cell can produce the protein, so that can be used by the human. All right, and also some human growth hormone that can also be produced by E. coli. So this is the pharmaceutical product of genetic engineering. Any questions so far? Brooke, where are you? I didn't see you respond, Brooke. Very good. Okay, so let's continue. So, for example, maybe you heard that. For example, some pharmaceutical company during this pandemic, they produce this. They try to produce this antibody, the COVID-19 antibody, so that they can inject the COVID-19 antibody for people to fight the virus. So this antibody is a protein. So the protein can be also produced through the gene cloning. All right. And so why gene cloning and PCR so important? All right. So because both technique can provide a pure sample of an individual gene, all right, separate from all the other genes in the cell. Okay, so they can provide a pure sample of an individual gene. And gene isolation by cloning or by PCR, these two procedures can give you the pure sample of individual gene. But for the PCR, it's a little bit more tricky because you need to know the sequence and also the size is small. But by gene isolation, you can identify the particular clone of interest, and they can be a large size, and you don't need to know the sequence well. Okay, so this is the standard procedure to show you the gene cloning. All right, as I explained previously. So you have a vector. This vector has been generated through some research. All right, and then they isolate this circular DNA from either the bacteria or from the virus, and then they use that as the vehicle to carry the piece of DNA, the gene of interest. So basically, you have this vector, and then you have the fragment DNA, and then the fragment DNA can come from the PCR or come from the restriction digestion, right? And then you have to cut the vector with a specific enzyme, all right? A restriction enzyme, which we will discuss later, right? Specific restriction enzyme. And then also you cut the fragment DNA with a specific restriction enzyme. And once you cut the same restriction enzyme, you open up this vector DNA, and so that you can insert, you can ligate this piece of DNA into the vector, so that you produce this recombinant DNA molecule. And then this DNA molecule, then you will introduce into the bacterial cell. And this process is called a transformation, all right, transformation. And as you may remember when you took the genetics, there are 3 different mechanisms that the DNA can be introduced into the bacterial cell. So this is first mechanism is called transformation, all right? So introduce the plasmid DNA into the bacterial cell. This is transformation, and this mechanism was discovered almost 100 years ago in 1928, the Griffiths experiment, all right? And so that's the first mechanism that you can introduce the DNA into the bacterial cell, called transformation. The second mechanism is called a transduction. Transduction, basically, you use the virus to carry a piece of the DNA from first bacteria, and then when the virus lyses the first bacteria, then they infect the second bacteria. Then during this process, the virus carry the DNA from the first bacteria to the second bacteria. And this process we call the transduction, because they introduce the DNA from another bacteria to the second bacteria. All right, so that's the transduction. And then what is the third mechanism? Anybody can tell me? to transfer a piece of DNA between. Yeah, very good, Muhammad. It's called conjugation. Yes, it's called conjugation. Very good. So that's the conjugation. When the one bacteria has appears, then they will contact the bacteria without the pillars, and then bring into the proximity, and then transfer the DNA to the second bacteria. So that's the conjugation. So remember, transformation, transduction, and conjugation. There are three important mechanisms for the bacteria cell to exchange the DNA. Okay, and then once you introduce this DNA into the cell, all right, and then of course the plasmid DNA will be multiplied, all right, and also during this process you also need to select the cell, select the bacteria cell that carry this recombinant DNA. We call these are the recombinant cell, and then of course there's a process to identify the recombinant cell that we will discuss later. Okay, so this is the general procedure for the gene cloning, and so this is the this is just to show you that the piece of DNA fragment, all right, that's from the you have a mixture of the many different fragments, and each one carry a different gene or part of a gene. And then if you want to identify this the specific function of the gene, then you need to clone this into the vector, all right? And of course, you by mix the DNA fragment with the vector, then they will have equal opportunity to be inserted into the vector, and then they introduce into this bacteria cell, and then you have this, and then you have the different colony. Each colony carry a different piece of DNA, and we call this as library. So when later on when we discuss the genomic library, that is how you produce the genomic library. Then you can take each one of these colony and to study the specific gene's function. Okay, and this is how you produce the gene library, and so you can study the gene function in detail. And for this example, it's just to show you that you have this genome, and then you try to clone a specific gene, all right? And then once you clone, and then you need to identify or select just this specific gene, and there are many different ways that you can then identify the specific gene. One easy way would be after the cloning, then one easy simple way is you just clone, and then you get the bacterial culture, and then you can perform the PCR to identify whether the vector, the recombinant cell, carry the specific gene that you want. That's one way. And another way is you can do the we call it a chromosome walking. Chromosome walking, that means you have to design a specific DNA fragment as a primer, and then you perform the hybridization to identify the specific colony that carry this specific gene. That we will also discuss later on in the class. Not today, I mean during the whole course, that later on we will discuss the more specific way to identify the specific clone. And then of course the PCR is the way to duplicate the specific piece of a DNA sequence that we will go into the detail later. So we are not going to spend time on the PCR technique at this stage, okay? But the point is that gene isolation by the PCR, you will need to know the sequence, all right? And then so you'll be able to perform the PCR to replicate, to duplicate this piece of DNA, and then you'll be able to perform the gene cloning, the gene isolation by the PCR. All right, any questions so far? Only 6 students respond. All right, so do you need to take a break now or you want me to continue? Okay, no break, that's good. All right, so let's continue now, all right? So the next section that we will continue now, it's called a vector for the E. coli. So as we know that during the gene cloning, all right, the first step is you have to use the correct vector. You have to choose the vector. No, that's not necessary. If you want to use a PCR, you can just use the genomic DNA. All right, use the genomic DNA, then you can perform the PCR and to get this pure isolation of the gene of interest that you want. All right. So the vector, okay, that's the first step. All right, the first step gene cloning. All right, so you need to have a vector, and then you need to have fragment. Gene isolation can be through the PCR cloning, yes. So this piece of DNA, that's the gene of interest. And then you have the vector, all right? So the vector is is a critical component for the genetic engineering, right? And then the vector in general, 2 different types of vector. One is called a cloning vector. The second type of vector is called expression vector. So the cloning vector, that basically is on the left side here, just to reproduce the DNA of interest. All right, that's the cloning vector, and which is very important because after you clone that specific DNA, then you will be able to study the function of that specific gene. Okay, and the second type of vector is called expression vector. It's a production of a protein from the cloned gene, like on the right side here. Okay, so today we will go through the cloning vector, and tomorrow we will go through the expression vector. Okay, so the vector basically central component of a gene cloning experiment. Okay, and the cloning vector need to be relatively small. Ideally less than 10 kilobase in size, all right? And there are 2 naturally occurring DNA molecule can be vector. One is called plasmid, that's from bacteria cell. It's a small circle of DNA, that's called plasmid. And second type of vector is called virus chromosome. Virus chromosome based they purify the chromosome bacteriophage and use that as a vector to carry the gene of interest. Okay, so 2 general type of vector, cloning and expression, and naturally occurring DNA molecule can be vector, plasmid, and virus chromosome. Okay, don't get confused. So the plasmid, the circular plasmid, circular DNA molecule, okay? So the plasmid, the basic features, all right? So that'll be, I highlight these three: selectable marker, origin replication, and cloning site. These are very important, all right? These are three most important features for a plasmid. And then also a size and copy number, conjugation, compatibility. These are all some specific feature for the plasmid, but these three are the most important one, okay, that I will explain one by one, all right, later, okay. So basically the plasmid, they purify the plasmid from the bacteria cell, okay, and the plasmid. So this is a typical plasmid map, and it's a little bit blurred, not very clear, but it's okay. So plasmid, all right, plasmid map. So number one, I say we need to have selectable marker, right? Selectable marker. Normally for the bacteria cell, they use the antibiotic resistant gene as the selectable marker. So why we need this selectable marker? Because as I mentioned in this diagram, so when you have when you clone a piece of DNA into the vector and then introduce into the cell through the transformation, and then when you mix the plasmid DNA with the bacteria cell, okay, so you have like hundreds, thousands, maybe 10,000 million DNA molecule mixed with million cells, all right? And then when you mix them, and then you apply the technique for the transformation, and then not every single cell will be able to uptake the DNA molecule. All right. So later on, I will also during the class that we will also learn how to calculate the transformation efficiency. Transformation efficiency. So basically, it's not 100%. Therefore, maybe only 20%, 30% of the bacteria cell receive this plasmid DNA, and right. And now you need to identify. So that means the other 70%, 80% of the cell, they don't have the recombinant DNA. And now, in order to identify the cell carry the recombinant DNA, which means we want to identify the recombinant cell, we need the vector to have a selectable marker that can help us to identify that cell carry this plasmid DNA. All right. The easiest way for us to identify the cell that carry this plasmid DNA is to use the antibiotic, all right, in the culture. So for example, if we add ampicillin in the growth culture, then we'll be able to identify the cell if the plasmid carry the anti-ampicillin resistant gene, the ampicillin resistant gene, then that can help the recombinant cell to survive and to grow, and then you will see the colony. That's how you identify the recombinant cell. And then question about the selectable marker. Do you all understand the reason why we need a selectable marker? Good. And then, so that's why the number one selectable marker is very important. And number two, original replication. Original replication, okay? Original replication is here, all right? Original replication. So the importance of the original replication is because when you introduce the plasmid DNA, the circular DNA, recombinant DNA into the bacteria cell, and then the bacteria, they will duplicate. And then as you know, the bacteria duplication is just binary duplication, so that they just produce they just duplicate. And then if the plasmid DNA does not have this original replication, therefore, when you duplicate a cell, all right, then only the mother cell carry the plasmid. Then the daughter cell will not get the plasmid. So during the duplication stage, they have to duplicate the plasmid so that when they produce they have a cell division, then the plasmid DNA can be equally distributed to the next generation, so that you can continue to maintain the plasmid DNA in the bacteria cell. Okay, so that's why you need the original replication to maintain the recombinant DNA molecule in the cell during the cell division. Okay, and then the multiple cloning sites. The cloning sites, all right. The cloning site is important is because if you have cloning site, then you'll be able to pick a specific site and then perform the restriction digestion and then to introduce a fragment of DNA into the vector. Okay, the multiple cloning sites, that mean multiple restriction enzyme digestion site, and the enzyme digestion that we will discuss later as well. Okay, and this is the actual a plasmid vector. All right, so that as you can see here, like here they say MCS, that means multiple cloning site. That means they have multiple restriction enzyme digestion site here, so that you will be able to introduce a piece of DNA into here or here. And this P1, P2, that means the dual promoter, all right, dual promoter. And then this ORI, that means the original replication. And this is for bacteria. And this 2 micron ORI, this origin of replication for the yeast cell, Saccharomyces cerevisiae. So this would indicate that this vector, right, is called a shuttle vector because this vector can be introduced into the bacteria so they can replicate in the bacteria cell, or they can replicate in the yeast cell. Okay, that's called a shuttle vector. And BLA here, that's the ampicillin resistant gene. Okay, that's the ampicillin resistant gene. And this is for the selection for the bacteria cell, all right? And also can be used for the yeast cell, but there's another selectable marker, all right, that can be used, all right, selection cassette here that can be used. So these are the typical the cloning vector that you can see here, right? Or expression vector. This is expression vector because they have this the FRAC tag or CMyc tag. These are for people to produce a protein so they can easily purify the protein, which we will also discuss later, right? And this is just to explain the great detail about the selectable marker for the antibiotic resistant genes, all right? So when you perform the transformation, some cell will have the plasmid, some cell without the plasmid. And then if you grow the cell in the media without any antibiotic, then both cells will grow so that you will not be able to identify which cell carry the recombinant DNA. And in order to identify, in order to purify the cell that carry the recombinant DNA, then you grow the cell in the presence of the antibiotics. Okay, so that's the reason why we need the selectable marker. And in this example, the RP4 vector carry the 3 different antibiotic resistant gene, like ampicillin, tetracycline, and the kanamycin. And then you can, based on the cloning design, then you can have different antibiotic to select the specific recombinant cells. So this is just to explain what are the antibiotics that we will skip, that you can read it yourself. And then these are commonly used antibiotic. Okay. And here's the working concentration. Like ampicillin is 100 microgram per milliliter. That's very standard concentration, all right? 100 microgram per milliliter. And then another commonly used antibiotic for the gene cloning is kanamycin. Is a 50 microgram per ml. Okay, 50 microgram per ml. That's kanamycin. And original replication, also just to explain the importance of the original replication. And the original replication sequences are generally high in A and T. All right, they are AT base pairs. Okay. And other factors that can affect the copy number. So the insert and the E. coli strain, the bacteria strain, right? And the growth condition and the culture inoculant that can affect the copy number. The copy number, that means how many copy in the cell, right? So for example, for this vector, PUC vector, in the cell can have like a 500 copy in one cell. For the PBR322, is like a 20 copy in the cell. The PET, the PET and PGen, these are all commonly used vector. Okay, and let's just tell you how many copy number that they can have in the cell. Okay. And the vector, so the plasmid, if it's a non-integrated plasmid, they will stay extra chromosome like this, all right, during the cell division. And then because of original replication, so they can duplicate the plasmid, so they can equally distribute to the total cell. And then the episome, that means the plasmid will integrate into the chromosome. Okay, so that's an episome. That's called episome. And conjugation. So we explain this conjugation that basically that can they have a conjugated plasmid, and then they can have a pierce and then transfer the and duplicate the DNA to the recipient cell. That's conjugation. And the conjugation, basically, that's the plasmid with the fertility, the F plasmid, right? But this kind of the cell is the plasmid, F plasmid, right? Or resistant cell, resistant plasmid, resistant to antibacterial agents. Or cold plasmid is code for the colicin, which can kill other bacteria. Okay, these are different type of plasmids. So we just go through the bacteria vector, and now the virus chromosome. So we can also use the virus chromosome as the vehicle to bring in the gene of interest. Okay, so the basic features, they have a simple structure like this head and tail, or like capsule. Okay, and the capsule here. Or they can infect the bacteria, all right? They attach, inject, and replicate DNA, and then assemble the new phage particle and kill the host, all right? So to kill the host, basically, it's a lactic life cycle. Of course, they also have lysogenic phage. So for the phage, okay, the infection cycle. So this is bacterial phage, all right? They have 3 steps. First of all, they attach to the outside of the bacteria, all right? And then they inject the DNA into the bacteria, and then they replicate the DNA, all right? And then they produce they express these DNA and produce the protein capsid, and then they package it, and then they lyse the cell and release the new phage particle. So these are the 3-step process for the phage infection. This is lactic cycle. And lysogeny, basically, this DNA integrate into the chromosome, all right? And during the cell division, they also duplicate this chromosome and also the viral DNA. And then, and when the timing is okay, then they will switch from lysogeny to the lactic cycle, and then they will cut this the viral DNA from the bacteria chromosome and become extra chromosomal, and then they can duplicate and then express and then package the viral DNA and then lyse the cell. That's right. So the typical one that people use is a lambda DNA, right? And it's a typical lysogeny phage. Okay, they can integrate the DNA into the host chromosome. Okay, and the gene organization, the lambda DNA molecule is like this. Then the DNA molecule is 49 kilobase pair long. It's very long, all right? So it's not very suitable to be the vector. However, we have to perform some extra steps to make the lambda chromosome DNA to be suitable for the as the vector, okay? And it can be a linear form and also can be circular form. That's because the end of the lambda DNA has a 12 nucleotide sticky end, all right? Like this 12 nucleotide sticky end. So that's why they can be a linear, or they can be we call this as a coside, all right? It's called cohesive end. We call this coside. All right, so basically they can form this circular DNA, or they can be a linear. So basically they allow the linear DNA molecule, okay, to be circularized inside the cell. All right, this is the necessary prerequisite for insertion into the bacteria genome. Okay, and also the coside can be the recognition sequence for the endonuclease that can cut the DNA, all right? And then for the package, when they cut, then you can package, okay? So this is the lambda chromosome. And then another one commonly used vector is from the M13, all right? This M13 particle, all right? They can inject DNA into the cell and then become a circular that single-stranded, and then they can make it double-stranded, they can duplicate and then express it, and then later on they are separate into the single-stranded and then circular and then package it, right, and then release it, okay? So that's the M13. And M13 size normally is less than 10 kilobase, so it's okay to be used as the vector to carry the DNA, okay? And it is easily prepared from a culture of infected E. coli cell and can be reintroduced by transfection, okay? And then for the M13, infection, all right? So these phages, all right, basically cell lysis never occur, okay? So that's the M13, that's the feature, the M13 phage, okay? So now we know that we have these three different type of vectors, okay? One is based on the E. coli plasmid, and then one is based on the lambda bacteriophage, and another one is M13 bacteriophage. Okay, that's from what we just explained. All right, the from the vector here, the vector. So we have two naturally occurring DNA molecule. Remember, so plasmid, that's from the bacteria cell, and then we can have virus chromosome. Right. So here are all the bacteria cell from the bacteria. And then we have the lambda phage, right? Virus chromosome, we have the lambda phage and M13, right? So you have to know we have these three different types of the cloning vector for E. coli, all right? For E. coli, these cloning vector based on the E. coli plasmid, based on M13 bacteriophage, or based on the lambda bacteriophage. Any question? Mohammed, where are you? Nancy. Neil. Oh, you respond already. Very good. All right, so let's continue. The cloning vector for E. coli, all right? So three different type of cloning vector for E. coli, all right? Based on the E. coli, based on the bacteria, M13 bacteriophage and lambda bacteriophage for E. coli. These are three very commonly seen vectors, okay? The first one is called PBR322, all right? A small size, 4.3 kilobase, and carry the two different sets of the antibacterial resistant gene. We have ampicillin, all right, this is ampicillin resistant gene and tetracycline resistant gene, okay? And original replication, these character here, PST1, PVU1, SCAR1, EcoR1, Hindi3, BNH1, and SAR1, these are the cloning sites, all right? And they have a special function that we will discuss later, okay? So this is the PBR322. And this is how they construct the PBR322, because from these two originally from these two different plasmid, right? So from R1 plasmid, they extract this ampicillin resistant gene introduced into the CoE1 plasmid, right? And that create this PSF2124 plasmid vector. And then for the R65 tetracycline resistant gene, all right, they recirculate the fragment, all right, they cut it, recirculate fragment to get this PSC101 vector. And then from the PSC101, then also they are mixed with the PMB8 that carry the original replication to produce the PMB9. And PMB9, then they are introduce this piece, the TN3, the ampicillin resistant gene into the PMB9, right? Introduce this piece into the PMB9 to produce this PBR322, okay? And then they rearrange the plasmid, okay? And then to give you this final version of PBR322. So that's, and then this PBR322, basically you have the piece of ampicillin resistant gene from R1, which is here, R1, and then tetracycline from the R65, which is here, from here, and then original replication, this piece, PMB1, from this plasmid vector, okay? So from these three different vectors, they make into this hybrid, the original, they make this a PBR322, okay? So that's the PBR322 vector. And then the second vector is a PAC8, which is a leg selection plasmid. and it has the insertional inactivation of the beta-galactosidase, and high copy number, and one-step screen, menin restriction site. That's PAC8. This is the PAC8. All right. So PAC8 menin restriction site. So as you can see, here we have the multiple cloning site. We have several different enzymes, all right, that can cleave the DNA at least different sites. And the ampicillin resistant gene, origin replication, LEGZ gene here. LEGZ, all right, insertional inactivation of the beta-galactosidase, which is this one here, LEGZ. And insertional inactivation, that means here we have multiple cloning sites, right? The LEGZ. So that means if we insert a piece of DNA into the let's say for example BANH1, if we insert a piece of DNA into the BANH1, then the LEGZ gene will not be able to produce the full length of beta-galactosidase. They will produce something hybrid protein, or maybe the translation will be stopped, will cause an early termination, all right? And so that you will not be able to express the LEGZ gene, and that's why it's called insertional inactivation. Okay, insertional inactivation. And one-step screen. Later on, we will explain what is the one-step screen, because the combination of the ampicillin resistant gene and the LEGZ that can help us to do one-step screen to find the precise recombinant cell we want. Okay, that's called one-step screen that we will explain later. So that's PAC8. Okay, and then the PGen3, the PGen vector is very similar to the PAC8, contain extra 2 promoter sequence and suitable for in vitro transcription of a cloned DNA. And these 2 promoter, T7 and SP6 promoter. So this is the PGen vector. Okay, very similar to PAC8. All right, so we have ampicillin resistant gene, origin replication, and LEGZ in the middle, and then we have 2 different promoter. All right, so you can clone the gene and inside here, and then you can induce the gene expression from SP6 promoter or from T7 promoter, and then you'll be able to study the transcription. All right, and then you can see the size of the vector is not big. So for example, PBR322 is 4,300 base pair, and then PAC8 is 27 base pair, 2,700 base pair, and then the PGen also 2,750. All right, so it's small size. So these are the vector based on the bacteria. Okay, purify. They purify this vector from the bacteria. Okay, so these are the E. coli based vector. And then for the M13 bacteriophage. Okay, so the important thing for M13 bacteriophage is they can produce a single-stranded DNA, and the reason you need a single-stranded DNA is because that you can use it for the DNA sequencing. That's why you use these M13 bacteriophage. And also they have symmetrical cloning site. The insert DNA can be excised from the recombinant DNA easily. So this is the M13 bacteriophage, all right? And this is how they construct the M13, all right? So they introduce, so for example, construction of the M13 MP1, they cut the M13 bacteriophage chromosome and then religate with a piece of LEGZ. So they introduce the LEGZ gene here, and then they can introduce the cloning site, right? by doing in vitro mutagenesis that can be done through the PCR. So you can introduce the a specific restriction site into the sequence. And so basically the M13 bacteriophage, they have this polylinker, and they are symmetrical, all right, symmetrical cloning sites. So you can see symmetrical. So from left to right and from right to left, that will be exactly the same, okay? So that's the symmetrical cloning site for the M13 bacteriophage. And this plasmid, okay, is a PMBO8, which is a hybrid plasmid, okay? So for this vector, then you can produce a single-stranded DNA up to 10 kilobase, okay? And for the Lambda bacteriophage, okay, the LEX-Z gene, okay, here, the LEX-Z, right, basically that LEX-Z gene produce the beta galactase, and then in the presence of the chemical called X-scale, the beta galactase will cleave the chemical X-scale and then produce the blue color. So we can use this to do the blue-white screen, which we will explain in detail later. Okay, that's the reason we have the LEX-Z gene, so that we can do the screening easily by looking at the color of the colony. Then you can identify which colony carry the recombinant DNA plasmid. Adele, what's your question?
Can you please repeat that again? I didn't quite understand that.
You mean the LEX-Z gene?
Yes, please.
Okay, the LEX-Z gene. Okay, for those vector, they carry LEX-Z gene. That's because that'll be easy for us to identify the recombinant cell. But later on, during the end of chapter 1, I believe chapter 1, we will have more detail. But just to summarize this is, so the LEX-Z, because they produce this beta galactase, and this enzyme will be able to cut the specific chemical. It's called S-scale. So in the culture, if we add the S-scale, right, and then that will produce the blue color, all right, and that's how we identify which cell is recombinant cell and which cell is not recombinant cell. Okay, that's the simple way to explain this. But later on, we have more detail. Okay.
Okay. Thank you.
You're welcome. And the Lambda bacteriophage. Do you see the difference? See even the M13, the size is small, right? It's less than 10 kilobase. And then for the Lambda bacteriophage, it's 49 kilobase. It's too long, so there's a size limitation. And also, because it's 49 kilobase, so long, then they have multiple restriction sites. This multiple restriction site doesn't mean that they are multiple cloning sites, because multiple restriction sites mean the same enzyme appear several times, and that will increase the difficulty for the cloning. All right. Therefore, the scientists need to re-engineer this Lambda genome. All right, to cut it. Number one, cut it shorter. Number two, eliminate several restriction sites. Brooke. So two things they have to to engineer the Lambda bacteriophage before they can use it. So number one, you have to cut this Lambda genome, all right, make it to smaller size so that you can use it. Number two, you have to eliminate several restriction sites from the genome so that you can use it, all right? So this is, for example, so they have to, so they cut the, so they re-engineer the Lambda bacteriophage by removing a big piece in the middle that's not essential to the Lambda bacteriophage, and then religate, all right, so that you can use it. Okay. In the meantime, also when you are religate this, and in the meantime, you can also reduce the number of the restriction site. Okay, and then based on the Lambda bacteriophage, then you can have insertion vector or replacement vector, right? Insertion vector, that means you you cleave the Lambda DNA and religate, become 35 to 40 kilobase long. And then you have Lambda GT, can carry up to 8 kilobase of the DNA. And again, here is insertional inactivation of CI gene, all right, that's different gene. And then Lambda ZEP2 carry up to 10 kilobase of new DNA, insertional inactivation, right? And the LEXZ gene. And then also you have replacement vector. You replace a segment of the DNA with one to be cloned. And then you, like Lambda EMBO4, you can carry up to 20 kilobase. And CosMe, that means CosMe a hybrid between a phage DNA molecule and a bacteria plasmid. That's called CosMe. Okay, that's CosMe. So it's basically a plasmid carrier host site and need a selectable marker like ampicillin resistant gene and the plasmid original replication. Okay, that's CosMe. And this example here, insertion vector. So we have insertion vector. So basically you cut a piece of non-essential region, right? Then religate, and then you have this site, cloning site, the CI gene, so that you can clone the DNA into here, Lambda GT10 or Lambda ZEP2, all right? And then replacement. So basically you have a cloning site, you replace this piece with another target gene, right? So that's these are based on the Lambda bacteriophage. So we have these two general types of the vector. One is the cloning vector, and one is the expression vector. And then for the cloning vector, we have this vector based on the bacteria and vector based on the virus. And the vector based on virus, we have two types. One is the M13 bacteriophage, and the other one is the Lambda bacteriophage. Okay, so that's the cloning vector. Any questions so far? Okay, do you need a break or you want to continue? No break?
Continue.
Continue. Okay, that's good. So so now we just finished the cloning vector for the bacteria. Now the vector for the yeast and the other fungi. Okay, other fungi, yeast. So because we sometimes use the Saccharomyces cerevisiae for experiment, and then of course then you need to have vector for the yeast and other fungi, and then for the yeast and fungi, and then we have 2 micron plasmid. 2 micron plasmid, 6 kilobase long, 70 to 200 copies, and also trophic mutant, and which we will explain in the next slide. Also trophic mutant, the 2 micron. plasmid here, 2 micron, okay? So the yeast 2 micron plasmid, okay? We have this REP1, REP2, all right? They involve the replication of the plasmid. Also, they have original replication, all right, as well. And the 2 micron. So what is the orthotrophic mutant? So that means orthotrophic mutant, that means if the chromosome has no LU2 gene, the LU2 gene orthotrophic, that means that it's commonly used for the yeast vector, because the orthotrophic, that means if you provide the media, that the media is lack of one specific amino acid, and then the yeast will not be able to survive. That's called orthotrophic mutant. Okay, so for example, in this cell, the yeast cell, lack of the LU2 gene, and LU2 gene basically produce the leucine. Therefore, if you want to grow this cell, you must grow this cell in the presence of the leucine. Then you'll be able to see the cell multiply. All right. If you grow the cell in the absence of leucine, then they will not be able to multiply. So that's why it's called orthotrophic mutant. And then the LU2, right? So if you use the LU2 as the selectable marker, right, and then you put this transform into the yeast, then only the cell carry the plasmid will survive. So the idea would be the same as the antibiotic resistant gene, okay, for the selection. Okay, and then we have the yeast episomal plasmid, YEPS. This is based on the 2 micron plasmid. It's a shuttle vector, right? Shuttle vector, that is what I just explained earlier. The shuttle vector, that means this vector can be reproduced in more than 2 microorganisms. So for the common example would be in this case is the episomal plasmid shuttle vector can be reproduced in the bacteria cell and also in the yeast. All right, so that's called shuttle vector. And then the example would be YEP13. The YEP13 can perform the homologous recombination, which we will explain in the later slide. The homologous recombination basically will help the recombination occur when this plasmid is introduced into the cell, and so that you can replace the chromosomal DNA, and which would be the example that I will demonstrate this week later on to show you how to generate a knockout cell. All right, you use the homologous recombination to replace the chromosomal DNA to knock out a specific gene. Okay, and then another type of vector is YIPS. It's yeast integrated plasmid. This bacterial plasmid carry a yeast gene. Cannot replicate as a plasmid. Need to be integrated into the chromosome. That's YIP. And then YRP, that's yeast replicated plasmid that carry the original replication. And YAC, that yeast artificial chromosome. And also there's also another one, it's called BAC, is a bacteria artificial chromosome. Okay, so these are the commonly seen the vector type for the yeast and other fungi. Okay, so why do scientists use the yeast vector? Okay, because yeast are eukaryotic. Yeast are the simplest eukaryote, and also it's the single cell eukaryote. And however, it still contains complex internal cell structures similar to those of plants and animals. Therefore, that's why people use the yeast to study the the for the research, so that we can connect the specific gene function to the eukaryotic cell. And also yeast can perform the post-translationally modified protein. So those post-translationally modified protein, for example, the histone acetylation. Okay, histone acetylation is a very important function for the gene regulation. Okay, and this particular function has been used for cancer treatment. So that's why it's also important to use the yeast to study. And then, unlike most other microorganisms, yeast have both stable haploid and diploid state. So you can use that for the genetic analysis, so that you can do the hybridization, and then to get these the heterogeneous or homogeneous of the genotype, and then you can study the specific gene function. Okay. And then yeast expression plasmid used in the lab typically contain all the necessary count to allow shuttling between E. coli and yeast cell. Okay. So that's why the scientists use the yeast vector. And then for yeast origin replication, okay, there's a specific origin of replication DNA sequence for the yeast cell. Okay, and for the Saccharomyces, we normally call it autonomous replication sequence, ARS, autonomous replication sequence. It's about 200 base pair sequence containing the essential ARS consensus sequence. That's 11 base pair motif, like this one here. That's 11 base pair motif. That's essential ARS consensus sequence, all right, to be recognized by the Saccharomyces cerevisiae to replicate the DNA sequence. That's the yeast original replication. Okay. And four main types that we just discussed. We have the integrated plasmid, okay, because they cannot replicate, they have to integrate into the chromosome and replicate the plasmid that contain the ARS, autonomous replicating sequence, and the centromere plasmid, YCP, right? That low copy number, right? And incorporate part of ARS along with part of a centromere sequence, that's SEN, right? And then also YEP, that's episomal plasmid. And the selection marker for the yeast, as we mentioned earlier, the yeast cell, normally we use the osteotrophic mutant. And then osteotrophic mutant, then we use the selection, there's also osteotrophic selection. And then, so that means we have to, that means the inability of the yeast cell to synthesize a particular amino acid required for the growth. Okay, that's the osteotrophic mutant. And these are commonly used selection marker for the yeast cell, like the His3, Uro3, Lys2, due to TRP1, all right? So for example, His3, that means produce a histidine, right? And then due to leucine, tryptophan. So these are commonly seen selection marker for the Saccharomyces cerevisiae. And so this is an example of YEPS, the episomal plasmid. And then you can see we have two antibiotic resistant gene and also one LU2, all right? LU2 to produce the leucine and original replication. So this is a typical YEPS, the episomal plasmid. And this is how they use the episomal plasmid for the cloning. All right. So you have the desired recombinant. All right. And also you have non-desired recombinant. And also you have the ligate vector. All right. So you have 3 different, yes. So let's say when you perform the when you introduce the DNA into the specific yeast vector, all right, and then you could have, let's say you have a mixture of the DNA, and then you perform the introduction, all right, of the DNA into the vector. Then you could have these 3 different results. One, they perform a self-ligation, so that means no DNA insert. 2, that has an insert, that the insert is a non-desired recombinant. And then you have another type of the recombinant DNA molecule that has the desired recombinant, all right? And then you can take these 3, all right, 3 different DNA molecule, different type of molecule, transform to the E. coli, all right, transform into the E. coli, and then you perform the selection through the. Can you all hear me well? Hadil, then that must be your assistant, okay? So okay, so after the transformation to the E. coli cell, okay, then you perform this antibiotic selection. Antibiotic selection. So my voice in and out. Nadia, what do you mean barely? Oh, I see. Okay. So basically, all right, let's continue. Oh, maybe because I pause. Sometimes I have to pause and then maybe drink the water because you said continue, no stop, non-stop. So I continue to talk. Sometimes I pause and drink and then I continue. All right.
No, I think it's my internet sometimes. Sometimes it's just like cutting a little.
Oh, I
But
see. Okay.
it's fine. Thank you for the recording.
No problem. Yes. Okay, so let's continue. So now, let's say this specific the insert that you like, all right, and it's insert into this tetracycline resistant gene, all right, and of course this vector has two antibiotic resistant gene, and then one is insert into the tetracycline resistant gene, and then so basically, and so when you transform this into a bacteria cell, right, and then in the presence of ampicillin, so only the DNA, only the bacteria cell that carry the vector, right, will be able to survive, all right, will be able to survive. So you see all these colony in the media, and then you purify the DNA from different colony, right?
Hello?
And then hello, yeah.
Hey, Professor. It keeps
Yeah.
cutting again. I can't hear you.
You cannot hear me?
Yeah, it keeps cutting. I don't know why. I logged out and then I joined the room again, and it does the same thing.
Oh, that'd be but the others, the rest of the class
Sorry?
can hear Well, so that must be your system's function. Yeah. So yeah, maybe her internet is not stable. And also maybe because this is the first day of class, and then the whole CUNY are using the Collaborate maybe. Yeah.
I can hear you now, though. I can hear you now. It's better now.
Okay, that's good.
Okay, thank
That's
you so
good.
much.
Yeah. Yeah. And then so you purify the DNA, transform the yeast, and then the yeast from the yeast, then you can through the autotrophic mutant, and then you by supplying the specific loose the amino acid or lack of amino acid, then you can identify the specific colony you want. So this is just an example, okay? So that you know that the reason why we use the autotrophic mutant to identify the specific recombinant cells. And then the homologous recombination. So this is the example that we will discuss in a great detail later on this week. So to produce the gene knockout cell. So for example, this YEP, okay, they can replicate independently, and then carry this LU2 gene, all right? And then so for example, this is chromosome. When you introduce this plasmid into the cell, then this YEP13, they can perform this homologous recombination by recognizing the homology of this flanking sequence on the vector and also on the chromosome. So they can replace, let's say, the mutated LU2 with the correct LU2. After the recombination, then you get they can swap these two pieces, all right? And that's called a homologous recombination. And this is the principle normally people use to knock out the gene specifically, okay? Then the integrative and the replicative plasmid that we just discussed. So the selection of the yeast vector based on these three different factors: transformation frequency. Okay, so it depends the number of transformer that you want to obtain per microgram plasmid DNA. And so, for example, YEPS normally has highest transformation frequency. So that gives you like a 10,000 to 100,000 transformed cell per microgram of DNA. Okay. And YIP, that's integrated plasmid, right? Less than 1,000 transformer per microgram. And then the second important factor is the copy number. So YEP and YRP has the highest copy number. So like 20 to 50 and the 5 to 10 respectively. And the stability, all right? So YIP has less transformation frequency, but the YIP has a very stable recombinant. Okay, that's because YIP can integrate into the chromosome, so that's why it's very stable. So these are the three factors normally people will consider to select the vector how they use. All right. And then YAC, that is the artificial chromosome. Okay, you can use this to clone a very large piece of DNA, right? And first thing you see that's different is the unique feature for the YAC. They have the centromere. So basically, the centromere, basically they can distribute the plasmid to the daughter cell equally, correctly. And they have the telomere, right? Telomere, that's where the replication stop, right? They have telomere here. And then they have, of course, the Euro3, the selective marker, and then they have the original replication. So that's a YAC, is the artificial chromosome. And then the cloning vectors for the higher plants. So we have 3 types of vector system used for the higher plants. So it can based on the naturally occurring plasmid of agrobacterium, right? Like the tumor-induced TI or RI plasmid to introduce new gene into the plant cell. So the TI vector, so natural occurring plasmid isolated from the bacteria, that's a soil plant pathogen causing a disease in plant. Okay. So basically, when the bacteria infect the plant cell, then the tDNA from the TI plasmid insert into the host chromosome. And then the tDNA called for the auxin hormone that weaken the plant cell wall, infect the plant, divide in large form of tumor. Okay, that like this, right? So we have TI plasmid, so the tDNA, so you can clone a piece of DNA you want and then introduce into the plant cell, all right, into the plant cell. And then they can insert the DNA into the plant cell. And this is what they produce the crown gall, the tumor-induced, like this, okay? So that's the vector based on the naturally occurring plasma agrobacteria. Or you have direct gene transfer using the various type of plasmid DNA. Okay, so you have direct gene transfer into the nucleus, or you can have transfer gene into the chloroplast genome, right? So, oh, it's not here. It's all right. And then also we have the vectors based on the plant virus, like Calimovirus and Gemini virus. So these are 3 cloning vectors for the higher plants, all right? And then this is a comparison of the DNA vector and then their application. So they have bacteria plasmid vector, right? Like a PAC8, the PBR322 or PGen, all right? For DNA cloning, and later on we would tomorrow talk about the expression vector. That's also that for protein expression, okay? And then based on bacteriophage, all right, bacteriophage, we have the lambda phage and also the M13, right? For the cDNA, all right, because they can also produce the single-stranded DNA and the expression library and Cosmi. And the BAC, the bacteria artificial chromosome, they can insert 300 kilobase to produce a genomic library, all right? Genomic library, that's easy for the sequencing. And yeast, artificial chromosome, YAC, that can have 200 to 2,000 kilobase for the genomic library. And then the TI vector, gene transfer in plants. So this is the comparison, the summary of the comparison of different types of the vector that we discussed. Okay. And then for the BAC, large low copy plasmid contain a gene encode for the F factor, not for the conjugation, except the large size of DNA insert, okay, 300 kilobase. Were used during the Human Genome Project so that you can make the genomic library. You clone the DNA fragment and then make the genomic library, and then you can perform the sequencing to to get the DNA sequence. That's BAC. And YAC is a miniature of the eukaryotic chromosome that contain the original replication, can clone as large as 200 kilobase to 2 megabase.